Good morning and welcome to our Alaska Collaborative on Health and the Environment teleconference on community-based participatory research in the Arctic, sources of environmental contaminants on St. Lawrence Island, Alaska. My name is Diana DeFazio, and on behalf of Alaska Community Action on Toxics, I'll be facilitating today's call. CHE Alaska is a regional partnership group of the National Collaborative on Health and the Environment. CHE Alaska aims to advance knowledge and effective action to address growing concerns about the links between human health and environmental factors. We've held more than 90 calls on a variety of topics, and you can learn more about our work and listen to MP3 recordings of those calls on our website at akaction.org. You can also find more information about the Collaborative on Health and the Environment at healthandenvironment.org. Today we'll be discussing an important research collaboration that began in the year 2000 on St. Lawrence Island. At the request of the communities of Gambell and Savoonga, and with support by the National Institute of Environmental Health Sciences, or NIEHS, Alaska Community Action on Toxics has been coordinating community-based research on the island for the past 15 years. We're thankful for the communities of Savoonga and Gambell for their support and guidance over the years, and to our research partners at UAA and at the University at Albany. We'll begin today's call by hearing from Merle Apasingok, who will tell us about how it came to be that the communities of St. Lawrence Island requested assistance from Alaska Community Action on Toxics in researching the sources of contamination on the island. Then we'll hear from our research partners at University of Alaska Anchorage and at University at Albany about results of recent exposure assessment studies on the island. Again, we want to acknowledge the role of NIEHS, our valued partners with the University at Albany and University of Alaska Anchorage, and the communities of Savoonga and Gambell for making this research collaboration possible. Now I'd like to introduce Merle Apasingok, a lifelong resident of Gambell on St. Lawrence Island. Merle is a community leader on the island and a member of the Protecting Future Generations work group, comprised of youth, elders, and leaders of Savoonga and Gambell who guide our community-based research. Merle and his wife, Rona, have been working for Bering Air, a commuter airline based out of Nome, Alaska, for the past 15 years. And Merle has been hunting since he was a child and is currently a registered whaling captain in the Alaska Eskimo Whaling Commission. Welcome and good morning to you, Merle. You may go ahead and begin.
Good morning, everyone, and thank you for this opportunity for representation for Gambell, and we do appreciate this. To understand more about how significant our involvement with ACAT and the model that has been used for some years now regarding environmental health and our human health, we got to kind of look back at two I would like to use two events in our human health history on St. Lawrence Island to get a better perspective. In 1878 and 1880 famine, that's well documented, we had famine and diseases, and to this day, on this side, some of it, as the elders have been telling it, is they do know some information from it, but they also note that it's a very mysterious event. But there was a huge die-off of our population back then. As we were, due to our lifestyle, we had a close communal family and community dynamics out here. And we were like sitting ducks when Yankee whalers came out here and introduced diseases unknowingly to our people. And the other one was the TB epidemic, recently being, our father's peers, our father's generation, which was mostly respiratory infections, TB being passed on through respiratory infections to the lungs. And what we're facing now is environmental pollution, contamination, meaning our food chain is compromised and our water is compromised. So those two events and pretty much the first part of our environmental contamination, these are what you call afflictions on the molecular level. And way back then, we didn't have any knowledge of, we being, the people and our public health authorities in the nation, we didn't have a lot of information and a lot of techniques or equipment that would do that would help us back then, as they would today if it happened today. Right now it's very rigid protocols established due to experience, similar experiences elsewhere. But even as we were a very vigilant people here on St. Lawrence Island, because of our isolation, the island and the community dealt with threats to the people almost immediately back then. But these human health events, as they were on a molecular level, we were unprepared to. We didn't know how to deal with them, as people were dying left and right. So our father, during the TB crisis, mentions that his age group were many, many people. And he was nearly close to death because of what the TB took a toll on him and others. So currently, our current challenges are environmental contamination of military waste, which is well established. I do want to make a point that even as the military left and discarded, they just up and moved out without remediation, and remediation had to take place many, many years later. I do want to say that our people were very, very patriotic. As the Cold War was heating up, our people would challenge any community as far as percentage of men, and young men and men, signing up to defend our community and nation. And that is quite well documented, with our people being involved with the Alaska Territorial Guard and later the U.S. Army National Guard. Through these, through the military, we were also defending ourselves. And but we all know that the aftermath, as our people were being afflicted with cancer, and cancer is usually on the molecular level. And we are right now, basically from perhaps Annie Aloha's work as a health aide, turned the corner there during her time to initiate some effort to improve our health out here due to many cancer our people are being afflicted with cancer. So we sought help because lab work, as we all know, requires a lot of diligence expertise and professionalism, and on top of that, we got to make some sense of what to do about it. So, very, very complicated problem, and I applaud ACAT for organizing a method, a model, if you will, to try to improve things, try to improve our surroundings a little bit. And what I'm really happy about this current effort right now is very inclusive. It includes the community, as we are a people that have been researched quite a bit. But what's different this time around, this research involves the communities. Our history is full of bargaining table meetings where we're just treated as accessories. But things are changing now, and instead of being accessories, we're equal partners, so to speak, where our contributions are almost equal to the powers that be, and our own people are involved in this research, trying to find more information about how we can abject change and make our communities' environment healthier. And we also appreciate the doctors and the professionals, our backstops of all this information heading their direction. Very complicated and very difficult to understand and to make some sense and present it to the public, general public, anywhere. So we're on a good path, even though as our challenges are great, and I want to encourage everybody to keep getting better at it, as we are still battling cancer and our human health. It takes us down to our roots as vigilant people. We're also vigilant on the molecular level, and that's what I appreciate very much. Thank you.
Thank you, Merle. And a reminder to everyone that there will be an opportunity for you to ask questions of our speakers towards the end of the call. Thank you. Now I'd like to introduce our next two speakers, Frank von Hippel and Sam Byrne. Both have presentation slides for today's discussion, so if you haven't already downloaded them, please do so. They're PDFs, and they are on our website on the homepage at akaction.org. The links are just below the title of today's call. Frank von Hippel was born and raised in Alaska. He received his AB in biology from Dartmouth College in 1989 and his PhD in integrative biology from the University of California, Berkeley in 1996. Since 2000, Frank has been a professor in the Department of Biological Sciences at the University of Alaska, Anchorage. Frank's research focuses on two areas: ecotoxicology and evolutionary ecology, both using freshwater fishes as study animals. And he'll be talking a little bit more about that. Frank's research incorporates molecular, organismal, and ecological approaches to solve problems in conservation biology and environmental health. A critical component of several of Frank's research projects is community-based participatory research with Indigenous people. Frank is an associate editor of the Elsevier journal, Environmental Pollution. And our speaker after Frank is Sam Byrne. He is a PhD candidate in the Department of Environmental Health Sciences at the University at Albany. A previous employee of Alaska Community Action on Toxics, Sam is now working to analyze the human health and exposure data from St. Lawrence Island. His research interests include the environmental causes of health disparities, health effects of persistent organic pollutants, and the social and cultural factors which influence exposure to toxic substances. Welcome, Frank and Sam. Frank, you may go ahead and begin.
Thanks very much for the introduction and for the invitation. So the presentation that I'll be giving, it starts out with a slide that shows a satellite photo of St. Lawrence Island and the Bering Sea, and the name of our NIH grant, Protecting the Health of Future Generations. So if you'll start there, you'll see that Northeast Cape is on the east side of St. Lawrence Island, and I'll be focusing my comments on our PCB results that we've taken using fish at Northeast Cape. And then Sam will talk more about the human results and also looking at Gambell and Savoonga. So please scroll to the second slide. And these are the questions that I'll be addressing in this short presentation. So first, at the conclusion of site remediation, is remaining PCB contamination due primarily to the formerly used defense site or to atmospheric deposition? So let me just explain this a little bit. Northeast Cape is the site of the most expensive cleanup that's taken place in Alaska. Around $123 million has been spent to clean up the formerly used defense site there. And so we're asking the question, okay, the cleanup is considered complete. The cleanup has been shut down as of last year. And so whatever contamination is still there, is that still due to that formerly used defense site, or is this due to contaminants coming out of the atmosphere? The second question, is the remaining PCB contamination biologically relevant for resident freshwater fishes? And we're using resident fishes as our model organism for human health. And therefore, the third question, do contaminant levels have implications for the health of people on St. Lawrence Island? So this is my collaborator, John Postlewaite, from last summer out on St. Lawrence Island, and we'll just scroll to the next slide. You can see that we use available materials for our cutting boards, and we're using two fish species as our model organisms at Northeast Cape. One is called the nine-spine stickleback, and the other is the Alaska blackfish. If you go to the next slide, you'll see that each fish gets divided up into lots of different parts. So we take some DNA from the fish in order to determine its genetic sex, and then we take various organs such as the gonads, the liver, the kidney, thyroid, brain, and some of those tissues we use to sequence RNA, so we look at gene expression with those tissues, and then match samples are fixed for histology so that we can look at how those organs have developed differently based on the contaminant load of the fish. So we also measure the contaminant chemistry of these fish. If you go now to the next slide, this is a picture of an Alaska blackfish, and what you can see here is on the x-axis the different PCB congeners. So there's 209 different PCBs, different types of PCB molecules, and they vary based on the number of chlorine atoms that are in the molecule. So there's very light PCBs that just have one chlorine, all the way up to heavy ones with 10 chlorines. So the lighter ones are readily transported in the atmosphere. The heavier ones are not moved readily in the atmosphere and therefore are of a local source. But this graph is asking is whether for each congener are the levels higher upstream of the formerly used defense site at Northeast Cape, or are they higher downstream of the formerly used defense site? And you can see that for the lighter ones, the ones on the left, there's no particular pattern whether they're higher upstream or downstream, and that's because these ones are deposited from the atmosphere. But all the heavier ones are higher downstream because these are ones that are locally sourced from the formerly used defense site. If we go now to the next slide and just look at the top panel, this is for Alaska blackfish only. And what it's showing is for each of the family of molecules for PCBs based on the number of chlorines. So the total monochloral biphenyls would be the ones with only one chlorine, all the way up to the deca, the ones with 10. And you can see it's asking the question whether the levels are significantly higher or lower upstream or downstream of the formerly used defense site. And for the PCBs that have 1, 2, or 3 chlorines, they're neither higher nor lower upstream nor downstream. Whereas for PCBs with 4 or more chlorines, they're all significantly higher downstream. So again, this is evidence that the PCBs are coming primarily from the formerly used defense site. The same data are shown at the bottom of the slide for nine-spine stickleback, but we have a smaller sample size there, so the pattern is not quite as clear. Let's go to the next slide. And in this one, we're looking at different sites along the Suki River, which is the river that runs through the formerly used defense site at Northeast Cape. And we have sites 2 through 14 on the x-axis. Sites 2 through 12 are all downstream of the formerly used defense site, and sites 13 and 14 are upstream. And we have total PCBs for both blackfish in blue and for stickleback in green. One of the interesting things about this slide is that even though these fish move around, we can still pick up their signature of PCBs based on where they are in the Suki River. So those that are where inlet streams come from the formerly used defense site, which is the red, every place is a red circle, have higher levels of PCBs than those who are at different spots. And if you compare the PCB levels of any of these sites to the levels that are in the upstream sites, you'll see that they're much lower upstream than they are downstream of the formerly used defense site. Let's go to the next slide. So our conclusion then is that levels of PCBs in the Suki River fish are still high, even though cleanup is considered complete. And contaminant chemistry reveals a mostly local source, that is from the formerly used defense site. So this leads us to the question, are these PCB concentrations biologically meaningful? Because if the levels are higher in the fish that are downstream of the military site, but they're not high enough to change anything, then it doesn't really matter. So we want to know, does it really matter? Let's go to the next slide. The first way that we ask that question is asking whether the hormone system of the fish is disrupted. And we ask this because PCBs are known to disrupt the hormone system of fishes and other vertebrates, including humans. Some PCB molecules are estrogenic, they act like estrogen, and those ones can be associated with estrogen-mediated cancers. Some are anti-estrogenic. Some of them cause hypothyroid conditions. So there's a variety of outcomes that may occur, and we've been looking at those outcomes. So go to the next slide, and what we have here are different sites along the x-axis, and then on the y-axis we have the concentration of vitellogenin. Vitellogenin is the egg yolk protein that only females produce because only females make egg yolk. So what we do is we measure the amount of egg yolk protein produced by males who shouldn't be making any, because males will only make egg yolk if they're exposed to environmental estrogens, that is if they're exposed to contaminants that mimic estrogen. And you can see that for the sites that are downstream of the formerly used defense site, that these are all males, that the males have significantly higher levels of vitellogenin. They're induced for vitellogenin production, whereas in our control sites, which are other places on the island, they're not induced for vitellogenin. Control sites is in quotes here because there's also atmospheric deposition of PCBs, so that we don't have a true control, but we can compare the military versus non-military, and we see that in the military site, the male fish are producing egg yolk protein, indicating that they're exposed to estrogenic contaminants. Next slide, we looked at thyroid hormone, and you can see that 3 of our sites downstream of the military site, that is SQ2, SQ4, and SQ7, that the fish are hypothyroid. They produce significantly lower levels of thyroid hormone than the other sites. So they're disrupted for both estrogen and thyroid hormone. And now if we go to our next slide, we can see that some individual fish also have disrupted development. So for example, the image that's on the upper right of this slide is showing the cross-section of a male testis that is highly abnormal in its development, as opposed to, and that's downstream in the military site, as opposed to a stickleback testis collected upstream, which you can see in the lower panel, which is a normal-looking testis. And we also find abnormal livers in many of these fish collected downstream of the military site. And if we go to the next slide, the next question is gene expression of the fish disrupted? That's me on the left and Heidi from ACAT on the right, and Jesse from Savoonga in the background. And next slide, what we found looking at whole genome profiles of gene expression is that expression of genes in the fish that are associated with DNA repair and replication, associated with proteases that break down proteins, associated with metal ion binding, and these symporters, which move around ions within the fish, were all differentially up or down regulated in the fish that are downstream of the military site. And in particular, a finding that's concerning is that those genes associated with DNA repair and replication were all down-regulated or produced at lower or expressed at lower levels in the fish that are downstream of the military site. So that's a gene expression profile that could be related to cancer. So if we go to the next slide is the last slide, our conclusion slide. I'll just walk through it. So number one, our first question at the conclusion of site remediation is remaining PCB contamination due primarily to the formerly used defense site or to atmospheric deposition. We found that most of the PCB contamination in the Suki River fish originated at the formerly used defense site. Second, is the remaining PCB contamination biologically relevant for resident freshwater fishes? We found that the ascensional fish in the Suki River show estrogenic effects, thyroid disruption, and altered gene expression. So yes, the contamination is biologically relevant. And finally, do contaminant levels have implications for the health of people on St. Lawrence Island? Our conclusion is based on the fact that fish and humans share the same hormone systems, and most of the genes underlying diseases in humans are the same genes underlying those diseases in fish. That's why we use fish as model organisms. So estrogenic effects that we found are associated with abnormal development and certain cancers in humans. Altered gene expression results are also consistent with higher cancer risk. The fish results therefore indicate that PCB levels in the Suki River are still high enough to cause health problems in people exposed to those PCBs through, for example, eating Suki River fish. And after Sam's presentation, if anyone has any questions on any of this material, I'd be happy to answer them.
Thank you, Frank. And Sam, you can just go right ahead, and then, as Frank mentioned, we'll have time for discussion at the end.
All right. So Frank talked mostly about PCBs and about military contamination, and I'm going to talk about some newer persistent organic pollutants with some slightly different sources. Before I go into that in detail, I'll just review some of the data on some of the human health data we have related to the military contamination. So if you go to the second slide, you can see that this is levels of PCBs in the blood serum from people on St. Lawrence Island. This is data from during the remediation of the site, and overall PCB levels on the island are higher than they are in the US on average, and that you can see that the individuals who spent significant amounts of time working near the FUD site or collecting food near the FUD site, the formerly used defense site, have the highest levels of PCBs. So there's some evidence that not only is the local environment contaminated, but people are also exposed through military contamination. If you move to the next slide, you can see very similar results, but this is for a few different groups of persistent pesticides, DDT, chlordane, hexachlorobenzene, and Mirex. And again, you can see that comparing the two villages and then people who spent time near the military site, the people who spent time near the military site, which is the column at the far right of each of these groups, tend to have slightly higher concentrations of these persistent pesticides. So we think that perhaps some of these pesticides may have been used at the military site and then left there. So Frank has done a really good job of looking at specific health effects, sort of digging deep into the fish biology to look at health effects associated with military contamination. And we also know that people are exposed. It's hard for us to do that kind of detailed health work in people. So we really have to rely on our knowledge that people are exposed, and also that Frank is really determining that these levels of contamination cause health effects to suggest that people are probably also experiencing health effects from this level of contamination. But now I'll move on to the next slide. Other sources of exposure. We know that there are a lot of different sources of exposure to persistent organic pollutants. Among them, as Frank mentioned, are global transport and distillation. So these chemicals travel through the atmosphere and are deposited in the Arctic. They build up in traditional diets. So when Arctic indigenous people eat whales or seals that live a long time and are high on the food chain, they'll build up high levels of these contaminants. And then there's the potential for indoor exposure, because the heating season in the Arctic is very long. There's potentially low air exchange in buildings, and the potential for buildup of pollution inside from commercial products that contain these persistent chemicals. So moving on to the next slide, I'll just give you a quick overview of the two groups of chemicals that we're looking at now. And these are perfluoroalkyl acids is the first one, also sometimes referred to as perfluorinated compounds, so PFAs or PFCs, they're usually called. They're used for a variety of commercial and industrial applications. both used, for example, as surfactants or firefighting foams on the industrial scale, and also just put into commercial products like raincoats, for example, cookware, some food packaging. There's evidence for a lot of different health effects of these chemicals. Among them are immunotoxicity, reduced fertility, and thyroid hormone disruption. And the principal sources are diet and also indoor dust and air. And moving to the next slide, which is about PBDEs. These are polybrominated diphenyl ethers. They're structurally very similar to PCBs, which Frank has been studying. They're flame retardants, which are added to a lot of different consumer products, mostly furniture and electronics and fabric. So they would be present inside homes in products. They're also persistent and bioaccumulative, so they'll build up food chains. And they've also been associated with a lot of different health effects, like cognitive and behavioral effects in children, anti-androgenic effects, so that's sort of competing with the effects of testosterone in a body, and then reduced fertility and also thyroid hormone disruption. And the principal sources for most people are animal products and dust. Moving on to the next slide, which is slide 7, I'll just give you our overall study design for studying sources of these chemicals in the people. So we know that both PFAAs and PBDEs are present inside homes and also in the food chain. So we know that there's potentially 2 sources of exposure. So we looked at indoor exposure. We took dust samples to measure the level of these compounds in dust. We took environmental samples, the stickleback that Frank was talking about, sort of as a proxy of what is building up in the food chain, so we can try to make inferences about what's in the diet. Then we took samples of human blood serum to look at the levels of these compounds in blood. And we also looked at thyroid hormone concentrations in blood. I won't get into that today because we're not really done looking, done researching the relationships between the thyroid hormones and the chemicals, but that is definitely on our list of things to do. So today I'll just be focusing on exposure. So moving on to the next slide, beginning of the results. Here you see the concentrations of 6 different PFAAs in blood, separated between men and women. You can see the concentrations from St. Lawrence Island in blue and from the U.S. population in red. You see that PFOA and PFOS, which are on either side of the graphs, are present at the highest concentrations in the U.S. general population, and they're the most commonly used PFAAs. They were put in the most products, they were produced in the highest volume, and so it makes sense that they're present at the highest concentrations in the U.S. population. They're also present at high concentrations on St. Lawrence Island, which makes sense because they're high production volume chemicals. But also on St. Lawrence Island, we see that PFNA and PFUNA appear higher on St. Lawrence Island than they do compared to the U.S. population. So if you look at those bars, for PFNA and PFUNA, the blue bars for St. Lawrence Island are a little bit higher than the red bar. So it seems like this might be a case of a unique exposure, that these chemicals might be coming from something about St. Lawrence Island that's different from the U.S. general population. And we think probably that's dietary. So we know from other research that those 2 chemicals are present at pretty high concentrations in marine mammals. And so those might be coming from marine mammals. So moving on to the next slide, here we have relative concentrations of these chemicals in dust and in serum, so sort of comparing what we find in the dust in homes to what we find in people. You can see that the major PFAAs in blood are PFOA, PFNA, and PFOS. And again, that makes sense. PFOA and PFOS are the ones used most. It makes sense that they would be building up. PFOA and PFOS are present in dust at pretty high concentrations too. But again, you see that PFNA seems to stick out in that it's high in the blood concentrations and it's low in the dust concentrations. The same is sort of true for PFUNA, so it's present at a higher relative concentration in blood than it is in dust. Again, suggesting that these compounds probably aren't coming from dust, but maybe coming from other sources such as diet. And overall, the absolute concentrations of PFAAs in dust were pretty low compared to other studies from around the world. and there's not a very strong statistical relationship between these two, between the chemicals in dust and the chemicals in blood. So again, we think that dust might be a minor source of these compounds, but it seems like there's another strong source, which we believe is diet. So moving on again, slide 10. Here we have a similar graph for PBDEs, so these are the flame retardants. Again, St. Lawrence Island is in blue, and the U.S. general population is in red. So this is just a subset of the many PBDEs we measured. We measured 40 or so. This is the major 6, so the 6 that were present at the highest concentrations. And these are also the major PBDs that were produced worldwide. So 47 and 99 and 100 are the main components of the penta PBDE mixture. And so that was really one of the most commonly used mixtures. It began to be phased out about a decade ago and was replaced with Deca, which is PBD 209, which you can also see is present at pretty high concentrations. You also can see that PBDE 153 looks like it's present at sort of a similar level to the U.S. general population. Most of these compounds are present at slightly lower than the U.S. general population, or about the same. And PBDE 153 looks like it's closer to the same. And that one is really it's present at pretty low concentrations in the sort of technical mixtures, but it's very bioaccumulative and lasts a long time. So it just tends to stick around in people, regardless of how they're exposed. But overall, you can see the PBD levels on St. Lawrence Island look like they're a little bit lower, but not a whole lot lower. And this is sort of notable because the U.S. general population has the highest levels of PBDEs in the world, and so compared to other countries, PBDE levels in the U.S. are really quite high. And so having levels that are similar on St. Lawrence Island, even if a little bit lower, is notable because it suggests that the people on St. Lawrence Island really are exposed to PBDEs at a similar level to the U.S. general population, which is actually quite high. So where do we think PBDEs are coming from? If we move to the next slide, just slide 11. The major PBDEs in dust, which are 47, 99, and 209, are the most highly used in commercial products. That makes sense. They're put into products, they break down, and they end up in dust in homes. And they also tend to be the ones that really show up in blood, which suggests that dust might be a source. The relationships statistically are much stronger than they are for PFCs or PFAAs. So it looks like the overall concentrations in dust are pretty high, and the statistical relationships between dust and blood are pretty strong, which suggests that the dust is a source of PBDEs. We think diet is also probably a source, especially for PBD 153, which tends to be really bioaccumulative and persistent, so it tends to show up in the food chain. And to a lesser extent, that's also true for PBDE 47, which is also higher in blood than in dust. It also tends to accumulate and is one of the most common PBDEs that you find in the food chain. So the relationships between dust and blood for PBDEs are much stronger than for PFCs, and we think that dust is probably a source of these compounds, although diet is also probably a source as well. So moving on to slide 12. So this is just a comparison of the levels of PBDEs and PFCs by village. So this is comparing Gambell and Savoonga, the people who live in Gambell and Savoonga. And I really wouldn't have given this slide a second thought. I made it pretty early before I started looking at Frank's fish data. But it shows that PBD levels are a little bit higher in Gambell than they are in Savoonga. And so if you move on to the next slide, you can see why that might be an interesting result. So slide 13, these are concentrations of PBDEs in Stickleback. And each data point is an average for about 10 fish. And so so this is data that shows the PBDEs in these different locations on the island. So you can see all the way to the right, you have levels of PBDEs at the FUD site, so at the formerly used defense site, which are pretty low. They sort of hover about 1 part per billion, and they're very similar to the levels found in the Tapi River, which is a more distant river, not very close to the FUD site, and also not very close to either of the villages. So it's sort of being used as a reference site. So you can see sort of across the board there, the levels of the major PBDEs and the total PBDEs are pretty low at the FUD site and at the distant river. But the levels in Troutman Lake, which are very close to the village of Gambell, are exceptionally high. The levels of PBDEs here, you can see the total PBDE levels in these composite Stickleback samples hover about 1,000 parts per billion. And that's comparable to what you would find in pilot whales, for example. So animals which are much higher on the food chain, predatory animals, animals that live a long time and build up these chemicals over time. And it's hard to really overstate just how high these are. They're really, really outside of the range of what you would expect in Stickleback that aren't subject to a really strong source of contamination. And we think that that source of contamination is probably the landfill, so chemicals leaching from commercial products and things that have been thrown in the landfill. And so we think that potentially there is a source of these chemicals that we really didn't expect when we went into this study, which is basically the landfill for the village. And we don't know to what extent this could be a source for people. It's clearly a source for the local environment, but we don't know to what extent people would be exposed through this route. So moving on to the next slide, which is slide 14. This is a similar graph, but this is for PFAAs. And again, you can see at the FUD site, overall levels are pretty low, and similar to those that you would find in the Tapi River, which is a reference site. And again, in Troutman Lake, which is near Gambell, the levels are very high. Again, the levels of PFAAs are similar to those that would be found in large predatory whales like beluga whales. And so these are really outside the range of what you would expect if these fish were only being exposed through global transport. This trend doesn't really show up in people. And again, we're not sure to what extent this contamination that we think is probably coming from the landfill is a source for people, but it's something that we would really like to look into. And overall, the Stickleback data really mirrors the human data pretty well. Both for PBDEs and PFCs, the same compounds show up, the same major compounds show up in both species, and it really suggests that Stickleback are a really great model organism for measuring the exposure to these compounds and also the health effects probably. So moving on to the last slide, what have we learned so far? Overall, we think that dust is a pretty important source of exposure to PBDEs. PBDEs are present on the island in the sentinel fish that we measured. So diet is also probably a source, but it looks like just like in the U.S. general population, dust is probably the single largest source for people. So that the indoor environment is very important for these sort of more modern persistent organic pollutants. Where are PFAAs coming from? We think that dust is probably a much more minor source for these. There are a few PFAAs that are higher on St. Lawrence Island than in the U.S. general population, and that really suggests a unique exposure source. And we think that's probably dietary, especially for PFNA and PFUNA. Those really show up on the island at higher concentrations than we would expect if the sources were similar to the general U.S. population. And again, the Stickleback data suggests that both PBDEs and PFAAs are accumulating on the island due to global transport. And overall, the same PBDEs and PFAAs are present in humans and in Stickleback. And the Stickleback data also suggests that there might be some sources of exposure that we hadn't really anticipated, including local landfills. So that's about it. I think it's time for questions.
Great, Sam. Thank you. Yeah, now we would like to open it up to questions and comments from our participants. So if you'd like to ask a question, please press *2 to unmute your line. Please also state your name and affiliation, and when you're done with your question, if you could be sure to mute your line again afterwards. And again, you can ask questions of any of our three presenters today.
Hi, my name is Rodney Gerritz. I'm a graduate student in the Rural Development Program up at UAF, and I have a background in environmental consulting. And I'm excited to hear about this this talk today, and just having learned about the sites and the projects. I've got a question for Merle. I'm curious if Merle, if you were involved in the Restoration Advisory Board for the sites near Gambell, and or the one that's still ongoing at the other site near Savoonga, and whether you could compare and contrast that experience with this community-based participatory research approach that the current research and that you guys just presented has taken.
Yes, I think for 4 years I was a Restoration Advisory Board member when I was involved with the Village Corporation, Siwokok Incorporated. And we had routine meetings with the Corps, facilitating the meetings with the island residents. And we were advisory. We noticed that we had limits to what we said and the effectiveness of our presentations. And when we voiced our concerns, the Corps took it as advisory, and which is still good because our comments were recorded and documented. And so, in reference to Northeast Cape, when I was a young boy, I went to go camping with my relatives from Savoonga, 2 summers in a row. And we were staying at a camp just west of Northeast Cape, and we would use our go over to Northeast Cape via ATV or boat regularly, as my family over there had camps at both sites. My experience over there, we were, when it was not cleaned up, I noticed that I was playing around in the engine room, which was deemed one of the most contaminated sites over there. I was literally stepping on goo in the engine room, just exploring, looking, just being curious. I remember being really saddened by the weathered gymnasium. I was thinking about how can we bring this gymnasium to Savoonga or Gambell, as we needed basketball courts. But we also, Eugene Tooley brought me several times to the site up there near the mountain, and he would tell me stories about how he worked and what he did over there. So as far as remediation in Northeast Cape, Eugene Tooley is the most knowledgeable of what the military did during the time they were there and what happened after they left. So he's literally a national treasure, because these 2 military sites here in Gambell and Northeast Cape, helped deter the adversary during the Cold War. So I always deemed Eugene Tooley a national treasure.
Thank you, Merle. Do we have other questions? Again, *2 to unmute your line and ask a question or make a comment.
Yeah,
Hi.
this is Steve.
Go
Sorry,
ahead.
we have two people, so yeah, one go ahead, and then we'll make sure that the woman who piped in after goes.
Okay. All right. My name is Stephen Seymour, and I'm with Green Cape, which is a group on Cape Cod, Massachusetts. And we have some the PFSs, or the perfluorinated chemical contamination, in Hyannis near a fire training facility, and also at a military base. And we've just started to treat the water in the Hyannis area with activated carbon filters. I don't know how well that is working, but at least there's an attempt being made there for the drinking water. The one question I have when I look in the information, it's really not clear to me if these are completely off the market now. I know they were getting phased out, and I knew that they were also in pesticides, but I didn't know, as a surfactant, I guess, and I didn't know which ones and if those are phased out, and if these are all completely not in use at this point anymore. The perfluorinated chemicals.
Yeah. So this is Sam. I can address that, I think. So the ones that I was just talking about, the sort of main ones that we really see in people, those are largely phased out, both in the U.S. And beginning to be phased out globally. But they're also being replaced with compounds that are really quite similar. One group of those is fluorotelomere alcohols, and those tend to be slightly more volatile, and so they get into the air better. And then they break down or are transformed into the older forms like PFOA once they're in the environment. And so the answer is kind of they have and they haven't been phased out, because sort of the parent forms like PFOA have been banned, but the newer forms, which can eventually degrade into those older forms, are still being used.
Okay, thank you. Someone else was about to ask a question. I'd like to invite that person to go ahead.
Hi, this is Patty from Alaska Community Action on Toxics. I am not a scientist, so and my biology classes were about 40 years ago. So I need somebody, please, to explain to me the difference between the stuff I'm familiar with as the harm that chemicals cause, which for us it was DNA mutations, and now we're talking about gene alterations in gene expression. And I wouldn't mind a little primer on what's the difference between those two.
Sure. This is Frank von Hippel. I'll be happy to address that. So there are some contaminants that are mutagenic, that is, that actually change DNA itself, and that's what you're talking about. When Rachel Carson wrote Silent Spring, those were assumed to be very common because radiation is mutagenic, and everyone was worried about radiation from nuclear tests and potential nuclear war. And so that kind of mindset carried over onto the chemical side as well. There are some chemicals that are mutagenic, but really the vast majority of toxic effects of contaminants is not through mutating DNA. It's through changing hormone systems, through changing immune function, and a lot of that is mediated through changing gene expression. So what changes in gene expression means is that the contaminants are affecting which genes are turned on and which ones are turned off. If you think about it, you have every single gene in every single cell in your body, but your cells are not all doing the same thing. The brain is doing something very different from the heart, which is doing something very different from the skin. So you have different genes that are turned on or turned off in each of these different organs. And so part of the toxic effect of many chemicals is to affect that pattern of which genes are turned on or off in different organ systems. And then that, of course, will change. What genes do is they code for the production of proteins, so that changes which proteins are produced, which then has a whole cascade of effects on different systems of the body.
Thank you. We have time for probably one or two more questions. So again, I invite participants to press *2 to unmute their line and ask a question or make a comment.
Yeah, hi there. This is Ali Hamade from the Alaska section of Epidemiology Environmental Public Health Program. Thanks for the nice presentations. I just had two questions. One, we see that there are PCBs in the fish, and Dr. von Hippel, you mentioned that these mean that the contamination has not been cleaned up fully. Could it be possible that because PCBs stay in the fish for a long time and they build up, could it be that these are from pre-cleanup remains? Also, was there a health risk assessment done to determine that consuming fish from the river would result in increased cancer risk? The third question, maybe for Sam, and when we're comparing different PFC concentrations in U.S. versus St. Lawrence Island, did we do statistical analysis on these? It just wasn't clear from the presentation. And thank you.
Thank you. Those are great questions. So I'll address the first two. So stickleback only live on average for 2 years, and so the fish that we collected in 2013, 2014, 2015 would have been born between 2011 and 2013 when site cleanup was terminating. So that's one of the reasons that they're a good model for current levels of contamination, because they're a short-lived fish. So they do reflect recent, as in the last 2 years of contamination. As far as the health risk question, yes, there was a health assessment done, but it wasn't done in the way that I would do it regarding Suki River fish, because the health risk assessment would not include any data that involved fish that are not consumed. So none of our data was included in the health assessment, and the only fish data that were included in the health assessment were from Suki River were Dolly Varden that were collected in the Suki River, primarily near the Bering Sea in the estuary. And those would be fish that spend most of their life in the ocean and therefore do not reflect the local contamination in the Suki River.
Great. And Sam, did you want to respond to the third question?
Yeah, so the question was, did we statistically compare the distributions of chemicals? And the answer is no, we didn't. And I think probably it's a good idea that we should. I think there's a couple of things that sort of limit our ability to do that. One is that we have pretty small samples of people from the island. And so on some level, I might not be sure that we could detect differences, statistical differences, even if they were there. But it would certainly be a good idea to look at that.
Great. Well, I just want to thank everyone again. We are at the end of the hour, but I want to thank all of our participants for calling in, our three presenters for your time this morning, and I'd like to invite people, if they do have further questions, would want to keep the discussion going, you can email me, diana@akaction.org, and I'll do my best to get your question into the right person's hands. Again, I just want to thank everyone, and I want to remind you that we do post recordings of our calls on our website, and we will send an announcement when that recording is available. So if you have any further questions, you can email me. You can also call the ACAT office at 907-222-7714. And thanks again to everyone. Have a wonderful day.